Welcome to today's presentation. It is a pleasure for us to guide you through the first chapter of The Compass of Pleasure by David J. Linden, Mashing the Pleasure Button. During the next 5 minutes, you will get an insight into the discovery of the pleasure circuit and questionable experiments that are connected to that. It all started with Donald Hebb, Olds and Milner accidentally discovering the reward circuit. By placing electrodes in rats' brains that stimulated the pleasure circuit and specifically the septum, the first step towards the accomplishment of discovering the pleasure circuit was made. The researcher replicated the Skinner's box with electrode stimulation. Thereby, a rat was placed into a box with corners marked A, B, C, and D. Rats received a mild stimulation when moving to corner A of the box and kept returning to the corner in order to receive that stimulation. This was also implemented in a different way by stimulating the rat's septum every time it pressed a lever. As a result, the rats did not stop pressing the lever and started neglecting other basic needs. The researchers proved wrong the theory of avoidance of pain as the main source of learning and instead substituted it by a model where behavior is said to be pushed forward by pleasure as well as pushed forward by pain. Researchers decided to implement the before-mentioned experiment with humans, more specifically to treat homosexuality. Therefore, electrodes have been implanted into certain brain areas of a 24-year-old homosexual patient. Stimulation to the septum resulted in experiencing pleasure. The patient was allowed to stimulate that area himself and could not stop, up to experiencing overwhelming euphoria. He even neglected his family, personal hygiene, and other factors in order to keep stimulating. In order to treat patients' homosexuality, stimulation of the septum was paired with material that displayed heterosexual interaction. The researchers invited a hooker to seduce the patient to see whether he would engage in sexual intercourse with a woman. He indeed slept with that woman and even got married to a woman after the experiment. This study represents a strong violation of privacy and dignity and is morally highly questionable.
So the question is, how do we actually experience pleasure? The neurobiological processes that might answer these questions are the following. When neurons in the brain's ventral tegmental area, short VTA, are active, they are sending information among long, thin fibers called axons, and thus impulses race from the VTA to axon terminals. The axon terminals at the endpoint of the axons can trigger a dopamine release when getting in contact with traveling electrical spikes in the nucleus accumbens. The release of dopamine can happen because the vesicle membrane of the membrane-bound blobs where dopamine neurons are stored is fusioned and, thus, releases the neurons which start to diffuse in the synaptic cleft and start binding to dopamine receptors. So, the VTA can send dopamine-releasing axons to different parts of the brain, which will be felt by the subject as pleasurable. In short, some experiences cause the dopamine-containing neurons of VTA to be active will be perceived as pleasurable experiences. So, what we've learned is how the activation of VTA dopamine neurons, which are also called the pleasure circuit, are able to actually produce pleasure. We can also see this in the picture of the next slide. The most important thing is that the experience of pleasure depends on the release of dopamine in the region of the ventral tegmental area neurons. As said before, when an electrical spike reaches an axon terminal of the VTA neuron, the release of dopamine is triggered. Once dopamine is released, it diffuses across the synaptic cleft. Drugs, for example, artificially increase the effect of dopamine released from VTA neurons. Dopamine-related drugs are highly addictive and can lead to ignoring basic needs such as food, hygiene, sex, etc., just as the rats in the 24-year-old patient did.
Thus, if an increase of dopamine release or blockage of dopamine reuptake leads to that state of total neglection in favor of what produces pleasure, what happens? If there is a deficit in the pleasure circuit, e.g., there is a diminishing in the number of dopamine neurons. This is what occurs with the neurodegenerative disease of Parkinson, due to the loss of dopamine-containing cells in the substantia nigra and VTA. This leads to the symptomatology where you suffer from tremors, stiffness, problems with balance and coordination, as well as from changes in your personality that make you more serious, introverted, and uninterested in seeking pleasure. Thus, if the problem is the loss of dopamine-containing cells, the solution might be the usage of dopamine agonists to compensate. Well, it's not that simple. If you use levodopa, that is a precursor of dopamine, you at least need to have the remaining dopamine neurons. However, if you use dopamine receptor agonist, you do not need to have surviving dopamine neurons, but you have the drawback of probably increasing the risk of gambling addiction and other risky behaviors. Thus far, we have seen how pleasure is produced in our brains, how these mechanisms work, and what happens when they do not. Nevertheless, do we know what is its purpose? Thanks to studies made with C. elegans, the underlying living being with that non-suspicious hair, we know that actions that satisfy basic needs, for example, following the chemical pathway leading to food in the case of C. elegans, produce and make us seek them. The difference with humans, I just googled humans and he appeared, and other complex living beings, is the complexity of the pleasure circuit. When a pleasurable experience is experienced, it is associated with the inner and outer cues in order to be able to repeat it, as well as some sort of quantitative value to decide if it's worth going for it or not.
To conclude, we tend to classify behaviors as virtues or vicious, depending on whether it's acceptable for society to seek pleasure through them or not. But the fact is that, for example, when we enjoy eating something we like, which is considered vicious, and when we have fun reading a book, a much more virtuous behavior, we are essentially using the same brain circuit. Therefore, should we look at pleasure just as the activation of the medial forebrain circuit and dopamine release? The answer is, of course not. The best kind of pleasure we get to experience often results from a combination of different information coming from various parts of the brain. For instance, we don't just enjoy the physical aspect of sexual intercourse, but also the feeling of intimacy and the boost of self-esteem we get when someone is attracted to us. While basic pleasure can be observed and manipulated through the medial forebrain circuit, we still have to investigate how this process is combined with other mental mechanisms to form more enriching, pleasurable experiences. Finally, we'd like to thank you all for your attention and let you know you can ask any questions you want. We'll try to answer the best we can.